All right, folks, we'll go ahead and introduce my colleague and mentor and friend, Kent Vlieger. He's the South Dakota NRCS State Soil Health Specialist, and Kent works to help producers and landowners across the state improve the health of their soil and resiliency of their operations. Kent has organized or helped organize a no-till community garden in Huron, where he lives, that incorporates cover crops, and many of you maybe even joined in that roundtable discussion on no-till gardening with him leading last night. So thanks for sharing your messages, Kent.
Great. All right, am I coming through loud and clear? Good, a few head nods from the back. Great. Okay, so like Tan said, my name is Kent Vlieger. I'm the State Soil Health Specialist with the NRCS in South Dakota. I currently live in Huron, and I have been there since 2007. I was in the field office. I had Beadle County and Sanborn County, for those of you that are familiar with South Dakota, as a district conservationist for a number of years, and that's kind of where I say I cut my teeth and did most of my kind of on-the-ground learning. I was fortunate to get to work with some great producers that were really forward-thinking on soil health practices. I was also fortunate to have a conservation district that now owns 2 demonstration farms for demonstrating all the principles that I get to talk about and work with every single day. So today we are going to talk soil life, soil biology. And my goal today is to make soil life and soil biology a little more relatable to the layperson, to the producer, to those of us that aren't necessarily doing research or doctorate students or doctors. So with that, I love visuals. I'm a very visual learner, so if you don't like pictures today, you're probably going to roll your eyes and say enough of it is enough. So what we've got here is just a spade full of soil that I collected this fall before freeze up. Okay, I know not everyone can see it. We don't have time to pass it around, but my people in the front row, does this look like good, healthy soil? It does, right? And how do you know it looks like good, healthy soil? Aggregation, right. Okay, you can see all the roots through there. You can see lots of aggregates throughout there, all the way down through the profile. Okay, it's nice and dark colored, right? Like we like to see. We know it's probably got pretty good organic matter. Okay, so where did I collect this from? What type of land production? Okay, everyone is guessing grassland, grassland. Another great guess. This is actually from cropland. Okay, what you're seeing on the top is some cover crops that were growing. It's actually a drought in this part of the state where it is this year, so they weren't much taller than this. This is from cropland. Okay, this piece fell apart in transport, so that tells you a little bit of something. Okay, what about this piece? What does that look like? It looks like a rock. It looks like a brick, right? This is something that soil scientists would call massive structure because there is no structure. It literally looks like concrete. Okay? This was collected less than 50 feet away, literally right across that fence, also in cropland. Okay, so what's the difference? Other than the visual, other than what we can see for aggregates, other than what we can see for color, what created this difference? Okay, tillage, the management practices is what made this difference. They're the same exact soil type. It happens to be our state soil, which is Hudak. There we go. This is a good educated crowd. All right, so management created this. Okay, so we got to that. We drilled down a little bit into the management, but why does the management on this side of the fence create this and the management on this side of the fence create this? Right, so you're getting exactly where I want to, Rick. We're getting down into the soil life. Life is what creates this. We can go from this to this with our management, or we can go from this to this with our management. Okay, we have that kind of influence on the soil. Okay, so keep this stuff in mind as we're going through all these slides in this presentation today. So soil life, what's in it for you? I would argue everything. And Rick hit exactly on kind of the crux of the matter is, and we're going to focus on this a little bit in some slides, is we want to capture as much sunlight as possible because that's how we push energy into our soils. Okay, so I'm not going to spend too much time on some of these slides because we're going to be a little short on time. We got 35 minutes or so to talk about this today. Dr. Chris Nichols did an excellent job discussing this slide yesterday. The point of this is to say there's a lot of life in the soil. We don't think about it because we oftentimes can't see it. Very few of those species that live in the soil we can actually see with the naked eye. So it's kind of one of those things that's out of sight, out of mind. But this is the quantity of life in healthy soil that we need to feed and keep active. Okay, this is a graphic. I know it's pretty simplistic. It's probably taking you back to middle school biology class. Kind of that circle of life type discussion. Okay, what I'd like to point out is, yes, this is simplistic. It gives a great example of all that interconnectedness between everything that's out there, between all the things that we can see. Oh, this isn't working. We got our larger mammals, what we can see, all the way down and back to the fungi and the bacteria, all the way back to our plants. And what I like about this graphic is here is it has the sun. Okay, so we like to say that circle of life. Really, it's not a circular system. Really, it's a high input system. And that high input is coming from sunlight. This system requires a lot of energy, but the only way we can get that energy into the system is through sunlight and through living plants doing what they do best, which is photosynthesis. So I think the previous speaker in the last room had a slide of even photosynthesis, the equation that's out there. So it's basically 6 parts of your carbon dioxide, 6 parts water, and it creates those carbohydrates. And then the waste is essentially the oxygen, which is what you and I breathe and need to survive. So it's actually a high input system. It's not necessarily circular. Once it kind of gets into the soil, it can be circular, but it still needs lots of inputs in the form of energy. Okay, so what I'm going to try and do today, as far as making it relatable for all of us to understand and bring home and say, okay, maybe I can make this system work. Maybe I can understand a little bit better what's going on in my systems. We have 3 basic functional groups that are in the soil. And what I like to do is I like to relate it back to something that I can see, something that I can really picture. So these 3 groups are ecosystem engineers. The best way to think of those is a lot of our trades workers. So construction, road workers, plumbers, electricians, anyone that's building and making things that we need to survive and that you can see driving down the road. Those are our ecosystem engineers. And oftentimes, when we were thinking about the soil, they're what we can see with the naked eye. So that's the earthworms, that's the millipedes, the centipedes. We got the wood lice, which is this little guy right there. My kids call them the roly-polies. You'll often see them. Sometimes if you have an older basement, you'll find them in there. So that's ecosystem engineers. Biological regulators. I like to think of them as law enforcement, as our healthcare providers. Even our lawmakers, you can think of them as the regulators. So really, they are responsible for most of the energy production and the cycle and the energy flow and nutrient flow that are going through there. The biochemical engineers. So think of them as the bankers. They're what make all those nutrients available to our plant roots. They are the things that we are focusing a lot on at this conference, and a lot of researchers are focused on. So that's our bacteria, that's our fungi. Those are the things that we can't necessarily see with the naked eye, but they are in great abundance in the soil. They're our highest populations in the soil. Okay, so we'll talk a little bit about each one of these functional groups. Ecosystem engineers, they're important because they build pore networks and they build aggregates. We all like to think about the earthworms. We all like to take a great big spade full of soil out and we like to look at the earthworms and say, oh, we've got earthworms, we've got a healthy soil. Okay, they are a good indicator for a healthy soil. Something to think about with earthworms is that there's 3 different groups, and we're going to talk about them here in a little bit. But these are the groups. This is the group that really is doing a lot of kind of the heavy lifting when it comes to breaking down residue and interacting with those larger pieces of organic matter that some of the bacteria and fungi can't necessarily break down on their own. Okay, so these are those earthworms. So this is a photo that I actually took from, I don't know if Anthony Bly's in here, I took from Anthony Bly's this past summer, or late fall. And what I'm pointing to there is a nice hole created by an earthworm. And we were in one of his fields, and you could find an earthworm hole basically every 18 inches. It was almost, you could almost measure it out. And it was interesting, you would find all that residue was pulled close to the hole, so they had all their food right there. And just the spacing of the food that was available for those earthworms, that's why it was about every 18 inches you could find a hole. It really was not hard to find. Once you found the first one, you could just kind of draw a circle around it and look for the next one. Okay, so things with earthworms, there's basically three types. There's the kind that we like to collect for fishing, the big night crawlers, and they usually create vertical holes that we see here. Those are the ones that most of us think of when we think of earthworms. There are the horizontal borers. And the horizontal ones are usually a little bit smaller and slimmer than those earthworm, those night crawler types. And they are usually within that surface horizon, the A horizon, and they're doing exactly what I just said. They're going horizontal through the soil. Most of our native worms that we have in our native systems are the horizontal borers. There's a lot of debate and still some questioning on the vertical borers, the big night crawlers, if they are native or not. We know there are a lot of them that are introduced. Don't think they're necessarily bad. There are some that are a little invasive. Minnesota's got some of those, the jumping worms. And so there's residue dwelling ones. We don't have necessarily a lot of those here in the plain states. They are more in the forest environment because they require a year-round constant litter, which is left by the leaf drop from the forest trees. Okay, the next one we're going to talk about is the chemical processors. This is the one that's kind of the soup du jour lately. So it's all the biological activity that is related to the bacteria and the fungi. We just had a lot of discussion with the previous speaker in the large room on some of that interaction, especially the ratios. This is one where I think this is where we sometimes listen to speakers and we listen to research. And if you don't really live in that world, I think sometimes your eyes glaze over because there's just so much information. And to be honest, there's a lot of information, and it's important information, and we need those researchers to be gathering that. But I also think we need to admit that we're probably never going to fully understand the interaction that takes place underneath the soil with all these different species. Okay, we'll fully acknowledge that there's probably hundreds of species we don't even have described yet. And if we don't have them described, we certainly also don't know exactly what they do and their function is in the soil. Okay, that's not the point of the conversation that we're going to have with you right now. The point is to understand how we can create better environments to maximize our biological life. Okay, so this is just a fun photo I took. Actually took this in 2019. So for those of you in the Dakotas, what was 2019 like? Wet, really wet. So this is actually from a grazing demonstration site south of Huron, and it's about the only time I ever wished that we had grazed the grass down to putting green height, because it would have made for an amazing photo. There was these puffball mushrooms. You couldn't take a step without stepping on a cluster of these. So this is what it looked like. You can kind of get the scale. There's my finger there for what they look like, and it was just so wet that all these puffball mushrooms were up and growing. And so this is a close-up of them. And this photo right in the lower corner is not related to it, but I'm just using this as an example. So this is a photo I took with just, it's actually a microscope that you can purchase if you want for your own personal use, and it connects with your phone. You can take photos and video of it. And I'm going to show you videos of some other fun things here pretty soon. But the point of this is a big blue stem root. Right there, the big white rope looking structure. Okay, that red line not showing up so good, that's the root. The circle there is, any guesses? I heard someone say it. Yep. It's mycorrhizal fungi, it's a hyphae coming out there. And this is really important. This is an aggregate attached to this root. And what you can't see kind of in the back, well, you can see just a little bit, there's hyphae. And hyphae's coming and it's essentially wrapped around that aggregate. So this is the start of the formation of those aggregates. Just for a size, so you can figure out the scale, from one end of the screen to the other would be 2 millimeters. So it is quite small. It's small enough where you can't see that with the naked eye, but this microscope zooms in just enough. Okay, the biological regulators. Like I said, these are something to think of if you're a visual person, like our law enforcement, our healthcare workers, and maybe even some of our lawmakers or politicians. And so what they do is they regulate populations in other soil organisms. So basically what that means is if a certain population gets too high, for example, then these regulators' population is going to increase because they're able to consume them, and it basically just keeps everything in check. And so a great example of these is nematodes. Most of us are familiar with nematodes. There are also, a lot of the other groups can fit in here. So this is a fun photo. And so this is mycorrhizal fungi that actually creates a loop. When a nematode goes through that loop, it essentially tightens on there, and then it has that as a food source. The fungi has that as a food source. So there's some really fun things that are out there.
So that's a microscopic snare trap, though, what you're saying.
Essentially, yes. That's a better way. That's a better way to describe it. I'm going to use that from now on. Thanks. Yes, just a snare trap. Okay, so I don't know if the back of the room is going to be able to see this. Hopefully you can. I'm going to hit play here in a second. And what I'd like you to look at is right there, kind of towards the upper left. Can you see that nematode? See how violently it's basically attacking that piece of residue? Okay, we're going to let this play for a little bit. So, the next slide I'm going to go to is why I'm glad I have you guys before lunch. Because the next slide is this nematode after it's full. Not in too much of a hurry anymore, is he? So you can see he's all full. It's basically a transparent body. You can see right through him. You can see all the organs, and you can see that he is full of whatever he was just eating. Okay, so nematodes. Most of us, if you grew up on the farm, if you heard the word nematode associated, was it a good thing or a bad thing? Bad. It was bad, right? Soybean cyst nematode would be a primary example. Okay, we need to kind of change our thinking on that, and I think most of us have. Most of us, if we're in this room, probably already have. Just about all the nematodes are predatory. Most of them consume the bad things that we don't necessarily want that are out there, and most of them are actually beneficial. We need them in the system. Okay, so we've talked about those three functional groups. Now what do we need to do in order to encourage them to be in our soils? Okay, so we're going to talk about managing for soil life. This is a side-by-side photo from a separate presentation I do, but it really fits this one as well. Okay, this is important because it doesn't matter which group I talk to. I could go to the We Love Tillage conference and show this slide, and I could ask them this question, and I would still get the same answer. Which one of these soils has more life in it? Right. It's obvious, isn't it? Okay, so what's interesting about this slide is they were taken just a couple miles apart. It was in Yankton County. Okay, so both of these fields had the same thing done to them this year. They had silage taken off of them. What's the difference? Cover crop. It's real simple. This is just before freeze up. That cover crop is maybe 6 inches tall. The producer is a little bit disappointed. And the only reason I know that is because he drove by when I happened to be taking a picture of his field. He was dry down there. But the point is, he has something growing there, right? He's covering the soil. He's taking that sunlight and pushing that energy into the soil. Okay? His point was he wanted to graze that this winter, but he didn't know if he had enough growth to do so. Okay, this is all management. They both did the same thing with their commodity crop, with the crop to make money, but it's what they did afterwards that makes the big difference. Okay, so this is where we talk about how that energy is pumped into the system. We've got the plant Photosynthesis is pushing those carbohydrates, those calories essentially down into the soil, those sugars. Okay, those sugars are then consumed by the microbes that are close especially to the root zone. And as those carbohydrates and the carbon gets cycled more and more and more and more, it gets broken down to a point where it gets to be that stable humus or humus. And when it gets to that point, it can last decades or centuries in the soil. So that's what we have in a lot of our cropland soil still today. Even if they've been tilled and tilled and tilled and tilled, they still have those organic matter percentages that here in the northern plains are still high because we're blessed with productive soils. Most of that organic matter that we now have left is that humus, the stable fraction that's not necessarily super active. And it's hard for all the soil life to get to and to start to break down and consume. Okay, so the conditions that we need in order to have soil life are these. It's kind of that Goldilocks zone, right? A near neutral pH, so somewhere above 6 to somewhere just a little above 7 is ideal. Moderate temperatures. Moist conditions, so not super dry. Aerated, and abundant food. Okay, what else likes those conditions? We do. Essentially all the same things we like is exactly what soil life. In fact, there's some things that are really similar. So like we like room temperature. I checked the thermostat. The thermostat is set at 68 degrees in here right now. I think it's fairly comfortable. Okay, it's that zone 60 degrees to 80 degrees is kind of right where a lot of soil life is really active and really happy, especially in the northern plains. Okay, so this is really important for us to understand. This is when in the northern plains, and especially in grassland systems in the northern plains, this is where we see the most activity. It's that early summer time frame. Okay, so why early summer? What does that meet? It meets all those requirements, right? So the soil has warmed up enough so it's not frozen anymore. Okay, we usually have reliable moisture earlier in the summer. June is typically our highest rainfall month in South Dakota. Okay. We usually have living plants that are in there, even in our cropland systems. Usually the crops are up and growing well into early summer. Okay, so that's what all the soil life needs. We've just created ideal conditions. And then you can see as summer goes along, it typically drops off. So what's the one factor that creates that drop off for the most part?
Moisture.
Moisture, right.
Lack of moisture.
Lack of moisture.
Plants are going from vegetative to reproductive state.
They- Exactly. That's another thing that we need to consider. The primary reason in the plains is we dry out a little bit. What Rick just touched on is the other thing. So when those plants are actively growing, they're taking a large percentage of that photosynthetic energy and it's getting pumped out of their roots. And we'll get to a slide here pretty soon if we get time to get to that. Anywhere from 5 to 20% of that photosynthetic energy is getting pumped out through the roots. It seems like a leaky system and it seems very inefficient, but it's important because exactly what Dr. Nichols talked about yesterday. It needs that exchange in order to gather things it cannot collect on its own. Especially when that plant is vegetative, it's doing a lot of that. As soon as it goes reproductive, it's not prioritizing that as much and it can start to pull from its own energy system. Okay, this is a great video. Hopefully a lot of you have seen this before. You can YouTube this, it's not hard to find. It's basically what we see happen when we have those soil engineers that are available in the soil. So the only difference between these two is this one has the earthworms and the wood lice and the mites in it, and this one has none of that soil life except for the fungi and the bacteria. So if you look closely, you can see the fungi growing on here, and you can see very, very, very slow breakdown. So that's the saprophytic fungi that's out there that starts to break down plant material. It's just on a lot slower process. This is over 15 weeks. And we're going to skip forward. You kind of get the point. This is what's happening when you have all of those functional groups that are in the soil. You get faster cycling of those nutrients. You get better nutrient transfer down into the soil, better energy transfer down into the soil. And that in turn provides better energy and nutrients to our plants, makes our plants healthier, which help to keep that cycle going. Okay. So those are the results of having all that activity in the soil. I'm going to pass around a core that was taken when we were doing a separate study this fall, and Just happened to come across this one. This is from a cropland system. The depth of this core that you're looking at here is just short of 8 feet. What I'd like for you to do is to look at both sides, look at the circular sides on either one. Just pass this around. Don't worry if it gets dropped or broken. I've got other ones. But just take a look at the pores that you can see in there. They are small pores. But that's the importance of having those engineers that are in the soil. And those engineers also include plant roots. The holes that you're seeing there were created by plant roots. Very deep down into the profile. Okay, can you guess what type of cropping system or what type of crop was in there?
Perennials.
Perennials, right. Specifically? Alfalfa, you're right. So, 8 foot deep. Corn root's probably not going to get down to that depth in that specific soil type, but the alfalfa was rooted down into there. That was from 8 feet deep, and that's all the deep we were going with this study, but if we would have gone deeper, we probably could have found it, and it'd be interesting to see how deep they actually did go eventually. Okay, so something else to consider when you're thinking about soil life is where to find soil life and really what conditions it really thrives in. So we call these things, we call biological hotspots. So we've got the root zone, we've got the litter layer, earthworm and root channels, pore spaces, aggregate surfaces. So something I'd like to point out is all of these are associated in one way or another with those 5 principles of soil health. Okay, I'm leaving out the 6th, the context, because we're not going to apply that to this today. But we got the litter layer. So which one of the principles of soil health does that relate to, or which two? Soil armor and minimize disturbance, right? So it's directly related to those two. Okay, thanks, Tance. Okay, we've got pore spaces, earthworm root channels, root zone. Those kind of relate to the following 3. So maximizing diversity within there. So that's plant diversity and animal diversity. We've got the earthworm root channels. That can relate back to disturbance. Okay, every time we disturb those soils, those channels are gone. Just a great example of how we can maintain that litter layer and cover on the surface. So there was a great presentation yesterday on interseeding and some different ways to think about doing that. This is a great example and one that I am keen on, and it's a really simple mixture of radish and clovers and annual ryegrass. Seems to work well, and it's a way to keep that biological activity going well into the fall when our commodity crop has stopped growing and pushing that energy back into the soil. Okay, earthworm channels. We talked enough about this. I'm not going to focus too much time on it. This is one that, let me get through here real quick. So if you look at a lot of soil textbooks, it'll tell you what that ideal soil looks like. And that's essentially 5% organic matter. That's that small portion right there. And then you've got the pore spaces that are filled with water and air, and that's 50%. And then we've got all the sand, silt, and clay that take up the rest. Okay, usually when you look at a lot of soils books, they'll say that's the ideal soil, what a healthy soil should look like. I'm here to argue that maybe we need to adjust those, and the reason I say that is because of a study that was done by Anthony Bly and David Karki and Sandeep Kumar and Navdeep Singh. Okay, this was done here a few years ago, and what they did is they took cores with plexiglass in different management systems, and those systems were conventional till with corn, soybean, a no-till production system, long-term no-till production system with soil health practices implemented, and then a really well-managed native pasture that hadn't been broke. So the point I'm trying to make is these are those same percentages on those management system types. So you can see there's essentially the pore space with water and air, with a lot of tillage, less tillage, cropland system with soil health practices. Here's the native range, never been broke, really well managed, well rotated. Look how much percentage of that soil profile is in pore space, either air or water. 67%. Maybe that's the standard we should be shooting for. Maybe that's really what we need to change our teaching on, especially when it comes to our soils classes. Okay, just food for thought there. Soil organisms physically stabilize soil aggregates. And so that's exactly what we're seeing here. You can see with the naked eye. The roots, they enmesh They can help keep those aggregates together. Mycorrhizal fungi do the same thing. They can create that mesh that's around the soil aggregate, and that's how soil life essentially builds its own home. It can take this and put it into this through that activity of life. Chemically alters or chemically keeps those aggregates together. So if you've looked at the slake test example that we have out there in the lobby, you'll notice one of the clods has been sitting there since, well, it's almost going on 48 hours now. The other one completely disintegrated in minutes. What's holding it together is, yes, to some extent, that actual physical holding of the roots and maybe the mycorrhizal fungi, but the thing that's holding it mostly together is the chemical process, the glues that are put out by that biological life, especially the glomalin that Dr. Nichols is famous for working with. Okay. Hotspot for chemical processors, those are the root exudates. So those are the bacteria. I think of bacteria as the greatest example. So they're the ones that like to live around those root zones. And they like to live around those root zones because that's where all those sugars are being pumped out. And the plant is putting out those sugars because they want to get something in return from that plant life or from that soil life. Okay, the rhizosphere, key organisms, saprophytic fungi are the fungi that are breaking down dead plant material and dead things. Mycorrhiza are the ones that are associating with our plant roots, and they really have that good give and take as far as nutrient exchange. Pathogenic are the ones we don't necessarily want, but hopefully if we have that good balance of soil life, those are minimized and kept in check. This is just a good visual. I'm just going to key on this one right here. This is the plant root for corn, and this is the plant root of corn with the extension of those mycorrhizal fungi. So basically just think of them as extensions. It's a way for that plant to go out and access more nutrients, especially the nitrogen and the phosphorus, that aren't necessarily readily accessible to that plant on its own. And there's that fact of the 5 to 20% of the carbon from photosynthesis is used to feed the below ground life. I've actually read a few studies where it could actually be pushed as high as 40% in some of our C4 plants. So those would be our warm season grasses specifically. Okay, I'm going to cruise through these. How can we manipulate the microbiome? The easiest thing to think of is those principles of soil health. If you've adopted all 5 of those, and with the 6th for context, you're going to be increasing your soil life home. You're going to be increasing the amount of food that's available for them. You're going to be increasing the environment or improving the environment so that they can thrive. Okay, below ground competition. We're not going to focus too much on this. This is just basically keep it into balance. This is a better photo of the snare trap. There you go. So there's some really fun things that happen. It's really a jungle down there. That's another way that I like to think about it. Okay, thanks, Tance. How do we feed them? We've discussed this a lot. We have to have living plants in order to feed them. Okay, just in review, biological hotspots, litter layer, rootworm and earth root channels, aggregate surfaces, and pore spaces. Those are the areas you're going to find life. They're just like us. They need a structure in order to survive. They need food in order to survive, and they need the right climate in order to survive. Okay, these are those soil health principles, and I like this graphic, and it's stolen from our National Soil Health Division, so thank them for that. It breaks our 5 principles down into a couple of categories, and one of them is basically protecting the soil, and that's what Soil Conservation Service was started and tasked with, and we've done an okay job with that over the decades. But that's minimizing disturbance and keeping the soil covered. It starts there. We have to stop erosion. If we can't stop erosion, we can't even think about building our soil. So think of those first 2 principles to begin with. Okay, and that's really kind of the whole no-till push over the past several decades has been those first 2 principles. Okay, but how do we go beyond that? How do we start building our soils back? How do we make them more resilient? How do we make them produce for us? That's these final three principles. That's maximizing our living roots. So having living roots in there from the time that ground is thawed in the spring all the way till freeze up and sometimes beyond. Maximizing diversity, and that's through crop rotation, through the use of cover crops and different animals out there, and then the integration of livestock. I'm not necessarily going to prioritize the 5. I think they all work together. You'll oftentimes see livestock as the 5th principle. I don't really care for that. I'm kind of a livestock person, so I'm really kind of partial to them, but I really think they help tie it all together. The producers that I've seen, that have really fully adopted these soil health systems and are really making them thrive and work almost always have livestock. The reason we have those livestock in the system is because that's how, think about how our soil is formed, especially in the northern plains. Large herds of livestock. That's how the soil was kept active throughout the winter, because those livestock can keep that biological activity really going along for those 3 or 4 months of winter when nothing else is really active and alive. Okay, that is my presentation. I think I'm probably close to out of time, but I've certainly got time for questions or comments, and I don't want to hold anyone for lunch. I'll be around for the rest of the day if anyone wants to talk to me one-on-one, but I do have time for a couple of questions if anyone has any real quick. Sure.
Do you have any resources for some of the pictures you've got up there so I can get the next generation a little bit?
Yeah, so there is actually a good one. It's called the Soil Biological Primer. NRCS has, field offices should have a copy of that for you to either look at or borrow. I also think you can order those online, but Soil Biological Primer, it does a really good job of kind of explaining exactly what I did here, and it gives great photos, and it talks some specifics on some species also.
If somebody wants one of those soil biology primers, we can just mail them to them. They just need to get the mailing address to us.
Sure, yeah, so contact your local field office and then we can get your information, so, yeah.
I think it's available just straight up as a PDF online too, if you didn't want the hard copy to misplace.
Yeah.